Hello there. Welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem. That's me. Today we will talk about concept of transformation. Concept of transformation. What is bacterial transformation and what is eukaryotic transformation? These two things we will study in this lecture. You see, this is what is bacteria I've drawn. Tentative bacteria where I've drawn with the few structure, not all of them. This is cell wall and plasma membrane and the circular genome and ribosomes. And it is, I call it as a E. coli because E. coli all the time is representing the bacteria because one reason is that it's well studied model organism. So I want to explain the concept of transformation with this figure, right? You see, this is what is the DNA in the environment. Bacteria, accidentally or incidentally, when it comes close to the naked DNA, then the DNA is enter into the bacteria. That process is called as transformation. Means the bacteria can acquire the or uptake the naked DNA from the environment, right? That process is called as a transformation. So not all the bacteria can acquire or uptake the DNA. Only bacteria which are competent enough, then they can uptake the external DNA molecule, right? So the some bacteria have a naturally have a high competency so that they can take up the DNA from the external sources. For example, Haemophilus influenza, that bacteria have high tendency of uptake of DNA from the external sources. But that's a naturally it is having high tendency. But to a certain bacteria cannot take up the DNA from the external sources. If that is the case, if that bacteria is treated with the calcium chloride, then that bacteria competency will increase and they'll take up the DNA from the external sources. So this is what is called as transformation. You see, the bacteria is come close to the naked DNA, and that DNA is enter into the bacteria. This process, uptake of DNA from external sources by competent bacterial cells, that process is called as transformation. Again, the definition is here. Uptake of DNA may be linear or circular molecule from the environment by competent bacterial cells is called as transformation. So usually transformation is part and parcel of the cloning. Whenever you dice the vector and insert and then ligate, and the ligation mixture is transformed into the E. coli to get the transformants as well as the recombinant molecules. So transformation is a regular procedure in the gene cloning, right? So E. coli strains are treated with the 100 millimole of calcium chloride, then that competency is as will be increased, and those strains can be used for transformation experiment. This is a bacterial transformation. But the same thing, the same concept cannot be applied to the eukaryotes because eukaryotic or animal cell transformation is different. Means, you see, conversion of normal animal cell into a tumor or cancerous cell is called as transformation. So the normal healthy animal cell is converted into the tumor or cancerous cell. That conversion is called as transformation. Normal cell, if they prone to some mutations, maybe somatic mutations, insertion, deletion, translocations, or amplification. So because of that, the normal cell lost its regulation over cell cycle, number of cell cycle it can undergo, right? Then the cell is converted to the cancerous cell. So normal cell have a fixed number of cell divisions before its death. But when it is because of the somatic mutations or germline mutations, there is an upregulation of oncogenes and downregulation of tumor suppressor genes. Then the normal cell is converted into the cancerous cell. That process is called as a transformation. See, normal cell which is accumulated, normal cell undergo finite or fixed number of cell division before its death. That's apoptosis. Whereas cancer cells divides infinitely and forms the tumor. Because the cancer cell means there is a upregulation of oncogenes which will break down the all the regulations of cell division. The cancer cells continuously divide. Uncontrolled cell division leads to the formation of more cell and the tumors, right? And also the cancerous cells are immortal; they won't die. So transformation in eukaryotic or animal cell means normal cell to cancerous cell is called as transformation, but in prokaryotes, uptake of DNA from the external sources by a bacteria, that is called as transformation. So to clear the differences and dissect the differences between the prokaryotic transformation and eukaryotic cell transformation, I have presented this small video. Hope you like it. If at all you like the video, try to subscribe my channel for regular updates. Okay. Okay, right? So if you have any doubts over the content and the concept, please write to me through the comment section. I'll get back to you as soon as possible. Bye bye. See you again with the one more video. Until then, bye.